we are Paula and Ben.
Today on Immunity Time, we're excited to welcome Professor Dr. Stefan Uderhardt. He leads a research group in the Department of Medicine 3, Rheumatology and Clinical Immunology at the Universitätsklinikum Erlangen. His work sits right at the intersection of immunology, imaging, and systems biology. He studies how our tissues actively regulate immune responses, especially the role of resident macrophages and stromal cells in keeping inflammation in check. One of his major projects, called NEXUS, is funded by the European Research Council. It's all about decoding the cellular networks that control inflammation in conditions like rheumatoid arthritis, psoriasis, and colitis. Stefan, we're really glad to have you here. Welcome to Immunity Time. Let's sip into science.
Well, thank you very much, guys.
It was a pleasure. So today I brought a Spanish tea from south of Spain, and it has a plum and mango, so it's a fruit tea.
Oh, really?
Yeah, and it's a green tea. So I hope we can, it's getting ready now, so I hope we can taste it soon.
So 100 degrees was okay then?
Yeah, I mean, for Spain, I guess, yes. Okay. Yeah, so do you want to tell us a bit about your path as a scientist?
Right. I would say it has never been fully linear. Yeah. So in fact, recapitulating everything I did was mostly by chance. It started with, so in Germany you have to do civil service, right? And you have to pick basically, or you have to decide what you want to do. And at some point it came up, okay, you go civil service or you have to go to the military, right? And I was like, I don't want to go to the military. Yeah? And my dad was like, well, what do you want to do? It's like, I don't know. Hey, if you don't decide, I'm going to decide for you, right? And I'm going to decide for you, you go to the hospital, right? And I was like, okay. So I joined the hospital and someone was like, hey, what are you going to do later? Right? It's like, yeah, I don't know. Hey, you have the grades to do medicine. Why don't you do medicine? It's like, yeah, okay, why not? So I got into medicine, yeah, and that's how I ended up, in fact, in the medical field, yeah, basically purely by chance. And then at some point you had to choose internships, yeah, and I couldn't decide. So it comes across that I'm very undecided, yeah. But the thing is that I only by chance, I think we let a pencil drop on a list of different clinics, yeah, and departments, and it ended up in medical department 3. Yeah?
Okay.
And this is where I am now, basically. So it's more like a fate thing, maybe.
Then you were lucky.
Well, it's like I went there and it was a nice environment. It was really something where you had to think about certain things, yeah, and it was encouraged to think more about pathology and, for example, underlying causes, molecular mechanisms and stuff like that than in other professions, I would say. Yeah. So during my time in civil service and hospital, I was working actually actively in the surgical department with them. So that was my first time where I was like, okay, you feel like a doctor because I was wearing a coat. Someone gave me a beeper and like a pager, and I got called into surgery. Yeah. Dr. Uderhardt, you're needed. And it's like, I was not even a student. Yeah. Okay. And I was like, I felt so like, but after a while it was kind of boring. Yeah? Yeah. So doing the same thing all over again. And that was the first time, like in the medical department 3 in immunology, I was like, hey, this is actually quite interesting. You can think about things. Yeah. And you have the time and you're encouraged. And that kind of what got me there. And this is when I first met like the director and my Dr. Vater, and this is how I got stuck there.
And why did you get interested in immunology?
It was something that it's like when you first get to know like cytokines and all that stuff, it's like a huge universe that is pretty much, and still is, like pretty much unknown and unpredictable in certain ways. And so I started my doctoral thesis then, and then at some point you realize, okay, you're actively asking questions and wondering about things that maybe don't have an answer at the moment. Yeah, so it was really creative thinking. Yeah, and you can explore a universe that it's still unexplored. Yeah, it still has basically, okay, on Earth we have discovered all, right? You don't have to sail across the world to discover new stuff, but here you can, and that was kind of exciting.
Kind of cool. Yeah. And so you started like with immunology, but right now you are studying like tissue resident immune cells like macrophages and so on.
Yeah.
How, like, did you have any mentor that kind of like guide you to this specific moment where you are?
We have, well, that's it, immunology. It used to be that adaptive, right? So B and T cells, but it's actually there's way more to it. Yeah. So when I first started, it was always like T and B and antigen presentation, and actually no one was really too much into macrophages. Yeah, they're kind of boring eaters that just sit around. Yeah. But in the department itself, there were a couple of influences. So first, my Doktorvater, Gerhard Krönke, who is now the director for rheumatology at Charité in Berlin. Actually also my best man at my wedding in Las Vegas, maybe in part 2. We were dealing with macrophages and innate immunity based on the models we ran, like the disease models. It was always something, inflammation. Yeah, it was not necessarily immunity, it was inflammation that we did. And so you automatically get into macrophages. So first we were dealing with macrophages that are recruited. Yeah, so you have 2 types of macrophages, in fact, in a human body or in a mammalian body. One part is sitting in your organs all the time. Yeah, that's the most prevalent, I would say, immune cell in a resting state in any organ. It's up to, there was a recent estimate with like 600 grams of your body is just steady state macrophages, depending on your body.
Yeah, actually, it's the first chapter of the podcast that we are talking about innate immunity. Like,
exactly. I mean, this is how everything starts, right? You can't get an adaptive immune response without the innate system. That's kind of a safety system, because the adaptive immune system can be so powerful that it basically kills you without control. And the innate system is definitely a layer of control. You can think about that, or think it that way. And so we were always dealing with innate immune cells, mostly recruited first, looking at how macrophages clear apoptotic cells, because this is what they're known for as big eaters. Macrophage, as my French colleagues say. So they are big eaters. That's what they were first described for, and that's what they do. So you can feed them and they eat. Yeah, but that has huge implications for. You can think of waste removal. So macrophages in your tissues are constantly sampling, taking up stuff, and removing dead cells, removing whatever shouldn't be there. So it's constant room cleaning, 24/7 service. And that is actually quite interesting, because when you think about it, okay, what happens when waste removal does not work? Then trash builds up and piles up, and in the end, it's just madness. And this is how we first basically started with innate aspects of inflammation, because if that waste removal doesn't work, you have, for example, waste lying around, and that gets necrotic, and becomes something that can activate the immune system. So you as a macrophage, you're basically trying to remove it before it goes bad. And if that doesn't work, then it might actually be the case that certain antigens get into the wrong compartment. All of a sudden, so for example, apoptotic cells, they are a source of your own identity antigens. So what we call autoantigens. And this is something that your immune system should ignore or even recognize as yourself and don't attack. But therefore, you need to establish immunological tolerance. And this can be done in the bone marrow, in the thymus, very early on during adaptive development and whatnot. Thank you. But it needs to be maintained in the peripheral tissues. And one mechanism that we described then in the end was actually that you need to that those antigens need to be contained in certain compartments, which act in a non-inflammatory way. And this is what resident macrophages do. For example, when you give, and the experiments were quite telling. When we had in a culture system, we had resident macrophages isolated from mice, and also like recruited more inflammatory macrophages. When you give dead cells to them, it ends up in the resident compartment. If you block that process, then it ends up in the inflammatory compartment, and that can then drive inflammation and can even induce immunity against the dead cells, so against yourself. And this was like, oh, brilliant. So this is actually quite interesting. So it needs to be contained. A potential source of damage, a potential source of autoimmune danger, I would say, maybe, needs to be contained in a certain compartment. And this was something very new when we worked on it and published on it. And this is kind of how I got stuck with the macrophages.
So it's not necessarily what is going on in the molecules and stuff. It's also the location that matters in terms of inflammation.
Yes, very much so. So, for example, location in the sense that our resident macrophages that sit in the tissues, they don't walk around. Yeah, so you have so-called migratory cells, monocytes, neutrophils, and whatnot. They are basically the mobile infantry and force. And then you have the stationary troops, which are employed in the tissues that are healthy, and they maintain that health. That's at least how we envision that whole process. And some areas, for example, the joints, where you, for example, have more damage and more like accidental damage, maybe like cartilage rubbing and friction and whatnot, there you need more macrophages, theoretically. And you actually, in fact, do. So it's the highest density of macrophages there, because you need more containment, because you have more potential damage and danger accumulating. For example, or another example would be then in the adaptive system, when you have, for example, when you mount an immune response and you have proliferation of B cells, for example, in a follicle, this is where you get massive cell death. And there you even have special macrophages which don't do anything at all, but taking care of that massive amount of cell death that occurs during the maturation process of an adaptive immune response and takes care of it and removes it. If that doesn't work, you get lupus. You get autoimmunity. Yep. And that is quite so. The waste removal is something that, like, everyone takes for granted, but it's actually super essential. And it always reminded me. So when you go, have you ever been in Manhattan?
No.
It's packed. So during the day, it's packed. It's a traffic jam everywhere. It's trash everywhere. And you're like, what? Jesus. Yeah. So how does it work here, right? So why doesn't that city just sink in trash? But during the day, there's no chance of removing waste. This is only possible at night at 4 a.m. Basically, the city gets cleaned and washed out, and so to maintain a certain level of homeostasis. And they have to go on strike like 1 or 2 days, and then the city is going to drown in trash. And the same is with the macrophages in tissues. We don't know in fact, specifically if there is a certain rhythm. So this is something that people work on. If, for example, if you clean at night, like it is proposed for the brain, that actually sleeping is some kind of a waste removal system. But it's all connected with the macrophages and maintaining a homeostatic state.
Yeah, because for example, because for the people that don't know, we already had lectures with you, and that's what we talked about it, right? So we had a moment of homeostasis.
Exactly.
So the body is like in steady state, and then we will have like a hazard, something that tries to harm us, and then we will have inflammation that will try to remove this problem somehow. So inflammation doesn't need to be a bad thing.
Exactly. So, this is something that Ruslan Mechedov firmly propagated, that notion that a certain thresholding is in place that maintains homeostasis. So that you basically have a homeostatic state where everything works as it should, like a physiological ground state. And anytime you have a deviation from that ground state, whatever it is, it can be temperature or whatnot, you have countermeasures to maintain actually that ground state. And if the countermeasures, the local countermeasures, for example, through macrophages, temperature might be a not good thing, but like, I don't know, something that can be removed, just very basic. And macrophages can deal with it locally, then you don't need inflammation. But as soon as that capacity is basically or the macrophages are out of capacity, then the only chance of removing that perturbation is through the immune system. So this is when you call in the guys.
Is it that there's more of a particular thing that the macrophages can handle, or is it the thing that they're handling is too inflammatory? What causes them to not be able to react properly and call in the immune system?
So it must not even be the macrophages calling in immune system. It's basically it used to be the idea that you have certain sentinel cells in tissues, for example, macrophages. But in fact, every cell is a sentinel. Yeah. I think we discussed that, right? So even fibroblasts can sense bacteria. Endothelial cells, they need to sense bacteria. They need to get activated and everything. Yeah? So then the macrophages are not necessarily the primary sentinel cell. Yeah? So if you, for example, let's say you have some kind of, I don't know, some immunogenic agent or so, and it can be removed by a macrophage, no one gets activated. Yeah. But if it's more than the actual amount that you can get rid of, then all of a sudden you get fibroblasts and everyone activated. Yeah. So even that capacity of just removing things through continuous sampling can be considered such a threshold. Yeah. So if you have, for example, one piece of LPS, one molecule floating about, a macrophage just eats it, or in fact drinks it, and then no one gets activated. So the constant turnover, it's like a whale fish. Swimming through water. In this case, it's not swimming, but it's soaking in any sort of solubles which are in the interstitial spaces. Yeah, and everything that is solved and is not large, it's not too large, gets basically taken up and ends up in particular compartments and can activate cells. Or if your threshold is high enough, then it won't activate.
In theory, anything that goes into your body can be eaten up by a macrophage?
Well, it's a size thing.
Yeah.
If something cannot be eaten up, so you have two choices as a macrophage. Either you eat it, and if you can down it, you crown it and build a wall around it. Rhymed only by accident. But it's actually what granuloma are. Yeah? So if you have certain things, for example, mycobacteria, which can't be killed, no matter what you do as a macrophage for some reason, or any sort of foreign body, plastic under the skin or something that is too big, for example, to get into a macrophage, the response would be to wall it off. Yeah, so that's the classical granuloma or foreign body reaction. Yeah, that can also be the case if a macrophage takes up something and can't digest it. Yeah, that can also happen. So macrophages can literally throw it up again, or they contain it, and then other macrophages, recruited cells, build around a wall around that cell. Yeah, so it's all about containment and compartmentalization.
So at the end, it's a matter of balance, right? Between
always.
Yeah. So it will be a bit of inflammation. It will be helpful. But too much, it will always be harmful.
That's always the case. So with inflammation, it's the thing. Again, its primary idea is to remove any sort of threatening danger, and allowing the system to get back to a homeostatic state or establish a new one. The problem with inflammation is that the regulation of its severity and kind of extent is the critical thing here. Yeah, so, for example, neutrophils, white blood cells that get recruited, they are super fast. They run through tissues. They do a dynamic behavior called swarming, which is super impressive. It's basically like piranhas around a dead carcass or whatever. Yeah, and similar to piranhas, they actually destroy the tissue. Yeah, so you as a neutrophil, you can't differentiate. You don't have that ability to differentiate between self and no self. Yeah, so you basically throw in the same chemical weapons against the bacterium or whatever is there. Yeah, so that your main focus is to sterilize a hotspot. And this is actually then what pauses. Pauses dead neutrophils and dead bacteria and dead tissue cells. Yeah, so this is causing damage. In a pimple or whatnot, this is on a small scale. But if you have something like this in the brain, if you have something like this in the heart, this is actually then what drives that collateral damage is actually what in the end can cause more damage than the primary insult. And that is also what the problem was with any sort of COVID, for example. Yeah? COVID, it was more the immune-mediated damage because of an excessive activation that caused the lungs to fill up and shut down. It was not the virus itself.
So at the end, the network of communication between the different cells is very important. And that is what you study in your project, NEXUS, right? Or
well, in this case, it is about communication, but it's more at the very, very beginning of inflammation. Yeah. So in terms of, for example, COVID or influenza cases, it's the excessive amount of cytokines that are being produced that further amplify and fuel circuits that then completely get out of control. So this is something where you, for example, have an advantage when you, for example, have less neutrophils. Yeah. So this was actually the case. I'm getting to NEXUS in a second. It's actually interesting. So we at first with COVID, we were a little bit afraid that our immunocompromised patients in the hospital, so patients with rheumatoid arthritis and whatnot, who get like anti-TNF therapies and whatnot, that they would end up in ICUs and get really into problems, but they didn't. You know, so in fact, turns out that certain immunosuppression actually was helpful, to survive a cytokine storm. Well, the cytokine storm wasn't too bad then. And this is what actually then in the third wave or something, on a regular basis, that certain cytokine blockade was introduced into the treatment of severe COVID cases. So it's the balance. And in such a case, the balance was completely off. NEXUS.
Yeah.
So with it is NEXUS is my ERC project, which deals with communication in cells, but on a more basic and also tissue level. So during my time at NIH, I did a lot of imaging. I did a lot of in vivo imaging, looking at different tissues, and also more in a volumetric way, so not in individual sections, but looking at entire cell clusters and cell agglomerates and whatnot. And then I actually saw that all those fibroblasts that sit in tissues. So tissues are compromised of fibroblasts, stromal cells that build basically some kind of a scaffold. The scaffold where all other cells are kind of nestled in. Among other things, for macrophages. And I saw that, okay, this is all interconnected. It's not just cells sitting next to each other, but they actually have interconnections, so physical interconnections, and it looks like a huge network. And you could actually follow that network. You can have one continuous line, I believe, from your pinky toe to your earlobe. Just following certain paths of stromal connections. So everything is connected. And what I was using back in the days was a calcium reporter system that shows you if cells receive communication signals, to put it that way. So if, for example, a B cell or a T cell gets activated through its specific receptor, then you see like a calcium flash. So calcium is a secondary mediator that is involved in all kinds of signaling and activities. And I used that system in the fibroblast in my tissue, and I could see, in fact, that without doing anything, cells were lighting up. And it looked like the brain, in fact. So I was like, hey, brilliant! I have something like brain activity in my stupid little peripheral tissue. So and that was then the basis of the idea that you have interconnection, you have network communication, and communication networks as a very fundamental organizational principle of how tissues work. So in this case, the idea is really like that fibroblasts are not sitting there by themselves, but they form, in fact, functional networks that allow them to communicate, cooperate, share metabolic traits, maybe share mitochondria or whatever. And this is now what we're studying. So the stromal network itself, but also the macrophages that are connected to that network. So you have to imagine you have a tissue-wide spanning network of fibroblasts, and then you as a macrophage, you come in later during development, and you kind of tune in. So you literally directly connect with that network and basically listen. And this is how you get very basic information about the tissue as a macrophage. In fact, when you block that communication signal, macrophages which will still be there, but they are more inflammatory activated. Yeah, so you need to be educated and imprinted by that tissue communication network to actually be able to fulfill your work and duty as a lawful macrophage. If that doesn't work, then you might be at a higher risk of developing inflammation where you don't want it.
You also talk about the imaging because you work a lot with imaging, right?
Yes.
And what is the advantage of like seeing the immune cells like there in life?
Imaging is a great way of is actually so. There used to be this the saying "seeing is believing." Well, it still is. Yeah. So if you claim something, show me. Yeah. Which is true for many aspects in imaging, and I still remember it very vividly. My really like my first imaging experiment where I was like, okay, this holy. It was a snowy December evening. It was actually the December the 9th, 2009, I believe. Yeah. So it was really like, it's like, whoa. So I did those experiments I told you earlier with apoptotic, with dead cells and recruited macrophages and not recruited macrophages. And we did some experiments beforehand looking at the distribution or the overall amount of dead cells that were taken up by macrophages using flow cytometry. Yeah, and there you could see, okay, there was no pattern. There was no pattern to it. It was just uptake. Yeah. But when I looked at the macrophage culture, you could clearly see that there was a differential pattern of uptake between the resident macrophages and the recruited macrophages. So that was the first time I was like, hey, this is actually this is something that at least back in the days we couldn't resolve using basic flow cytometry. So this was one of those aha moments where I say, okay, here imaging can actually make a difference. All of a sudden we see pattern, and by pattern I mean like spatial distribution. And this is something where imaging comes in, is quite critical. If you're interested in any sort of spatial aspects, you need to look at the tissue or at the cell or whatever. And this can only be done with imaging. And by imaging, I mean not only the microscopy that we do here, but that also extends to like spatial transcriptomics. Is also some kind of imaging.
You're not just looking at the cells under a microscope. You can also, with the imaging, for example, in the microscopes, you can take like a 3D snapshot of the tissue.
Yes.
Right under the microscope, and then you can see how things are distributed.
Yes. So the distribution of things, we established earlier that macrophages don't move. Yeah. So they are wiggling around and sampling, but they don't migrate. That means, in fact, that their position is fixed in the tissue. Yeah. So they, for example, macrophages are arranged in tissues in complex three-dimensional arrays or in matrix or whatever, right? And so their distribution is a product of the tissue anatomy. Yeah, and this is something you can only address with imaging, any sort of imaging. Yeah, you can take a section, but then you don't know what's happening basically below the section or on the other side or whatnot, right? And that's why we actually go for 3D tissue imaging, where you can basically, if you do it properly, you can catch a lot of anatomical features, vessels, nerves, and so on. So you can really describe the tissue that surrounds a certain macrophage compartment, and hopefully it allows you to extract certain organizational principles that you can then relate later with functional aspects. Yeah, to kind of understand why certain tissues are built that way. Yeah, so kind of a blueprint of a tissue.
Do you see the cells like alive? Are they not moving, but reacting to stimulus or something?
Yes.
Like they're alive.
Yes. So
like it's not like, for example, when you do, I don't know, immunohistochemistry, like you at the end, like
both. So certain things, for example, when you do certain methods of immunostaining, that can only be done on fixated samples. You can study, for example, organ anatomy on fixated tissues. You stain them with specific antibodies, which have, for example, fluorophores attached to it. You make them shine. and nicely labeled in a tissue. You can do that in 2D sections. You can do that in 3 dimensions. That's then a little bit more complicated. You have to make tissues translucent using something like optical clearing, which works quite well. So there are groups, for example, Ali Eturk in Munich, who made an entire mouse see-through. So then that generates like terabytes upon terabytes of data that you then have to find out how to analyze. This is more like the structural and organizational, I would say, analysis. So this where you can study how an organ or a tissue is built up. But we also do functional imaging, for example, intravital imaging, or for example, looking at macrophages, how macrophages interact with certain things, how macrophages respond to certain, for example, inflammatory cytokines. Again, here is something that we always want to achieve, is actually translating our images, either 3D or dynamic in vivo, in any sort of quantifiable metric, which can be actually quite tough, and sometimes you have to go new ways. That's why we're working a lot with the AI department here in Erlangen, and also with mathematics, with the mathematics department, and so it's actually quite interesting to try and extract more information out of your imaging than just say, look, it's more red. So away from the actual just qualitative interpretation to actually something like, okay, we want to bring imaging more towards like big data. Something where you actually can use Excel anymore. That's the data goal.
I don't know, it's pretty amazing. It's like something out of Star Trek, you just take a scan of this tissue and then you can quantify and qualify things about it.
I might
or the section of this tissue.
It just takes way much longer time. But in principle, yes, you want to actually understand how the tissue operates. You want to get at least a snapshot of the state of the tissue. And in fact, so macrophages, we use them as canaries in a coal mine. So it's, for example, in joints. So in joints where you can get arthritis, way before arthritis, the first kind of inflammatory stimulus leads to macrophages shutting down their sampling. They basically, you guys know those Christmas corals? When you go diving, you come close to them, they kind of retract immediately. So this is what macrophages also do. Certain macrophages in certain compartments upon certain activation. So just looking at a macrophage, we can tell its activation state just by looking at its morphology.
Now that you mention arthritis, because you have this project NEXUS to study the networks, and you also use it for arthritis, like rheumatoid arthritis. And rheumatoid arthritis is like a very common disease in old people, and not that old people too. Can you also like explain us a bit, like what is rheumatoid arthritis?
Well, rheumatoid arthritis is a autoimmune disease which is actually with increasing sensitivity and increasing healthcare capacities. It gets diagnosed more and more. In fact, it's an autoimmune disease directed against your joints, so to speak. So it has nothing to do with age, although you have certain age characteristics, but it's usually not your grandma with joint pain. So it's more like my age. So it's an antibody mediated, or oftentimes associated with autoantibodies at least, chronic inflammation of your joints. So in many cases you have autoantibodies, but that's not necessary. So somewhere the break of tolerance happens also at a level that we at least now cannot detect yet.
So we don't know why there is this break of tolerance.
Exactly. And also like you don't have any indicators for it. So anything we can measure in the blood, we have to know what to look for, right? So certain autoantibodies, we need to know that they are like what to look for. If we only select 3, then maybe there's a 4th one we haven't discovered yet. So right now we call it seronegative. And we don't know where it comes from, but very likely there's something going on that we just don't measure now.
But you can have certain autoantibodies without the disease, right?
Yes. So there are, for example, this wide class of anti-nuclear antibodies called ANAs, which at least at a rather low concentration is actually quite frequent in the general population. So just because you have certain autoimmune features doesn't mean that you have an autoimmune disease. Yeah, so certain antibodies like these, or for example, classic rheumatoid factor, so you can have those antibodies without ever developing an autoimmune disease. Yeah, so there's still this missing link between break of self-tolerance and then actually developing a disease from it. Yeah, and this is oftentimes, so you get a certain predisposition, for example, something genetic that leads to that break, but you need that second hit. Yeah, that can be, for example, an infection. That can be, I don't know, you took the wrong turn at the gas station, whatever. I don't know. Yeah, so a lot of environmental factors. Smoking can
Don't
cause
smoke.
exactly can cause certain things. Yeah, and so in regard to NEXUS, for example, in this case, the vision or the idea would be that. So NEXUS comes into place, all these communication networks in tissues would be before you get disease, yeah, or what happens after disease. Yeah, so the whole idea of NEXUS is also that the stromal communication is important to maintain a high inflammation threshold. So if your stromal communication works and your macrophages are in good shape, you can, for example, remove damages and potential danger molecules faster. Yeah. If that communication is abrogated, for example, if that connection between macrophages and the network doesn't work, you get a more inflammatory polarization and you're more prone to actually develop inflammation. Yeah. So question would be, for example, can we detect in tissues certain decimated network responses and therefore say, okay, this particular compartment is more prone to inflammation? Of course, we can't do that in humans prior to disease very often. So we have cohorts with patients that have autoantibodies but no disease yet. Yeah, but that's super tricky. So we focus more on in fact, looking what happens after we treated them. Because very often you have rheumatoid arthritis, you come to us, we kind of punch the inflammation down and with medication, immunosuppression and whatnot. And at some point you have to take it away because it can cause problems and it costs a lot of money. Yeah, and then it's basically, then you just wait. Yeah, and very often it's the case that we get an inflammatory relapse. Yeah, and one hypothesis would be, for example, at the tissue level, that it actually never fully healed. Yeah, that we have, for example, compromised communication networks. The macrophages are a little bit icky and can't maintain a high inflammation threshold, and that leaves the tissue basically prone to another attack. Yeah, and this is where I would say this whole NEXUS idea comes in for diseases like rheumatoid arthritis or psoriasis or whatnot. Yeah.
So at the end, with like rheumatoid arthritis, we are trying to shut the inflammation, right? And the typical, like if I go to the doctor with rheumatoid arthritis, the first thing that they will give me is steroids.
Depends on your case, yes.
Okay.
Yeah. That's the thing right now in rheumatology and any related like inflammation medicine field, that our main goal at the moment, and it's mostly everything we can do, is actually punching down the acute inflammation and hope that it won't come back.
Okay.
Yeah, and that's why it's so great that we in Erlangen were actually able, in the case of a handful of lupus patients, to actually achieve an immune reset. Yeah, by this whole CAR T cell story, that we got rid of the bad B cells in the body that produce the autoantibodies. And in those patients, for example, this was the first time that we could actually heal a chronic autoimmune disease. So we don't fully understand how and why and who would benefit from it, but this was a curative approach. Yeah, because usually we just handle the inflammation itself, but not the underlying cause. Because most of the times we don't know where it comes from. We don't know where RA, rheumatoid arthritis, comes from.
So we remove the symptoms, but we still have the autoimmunity behind.
The autoimmunity will still be there. Yeah. So no matter what we give the patients, the antibodies, for example, they remain untouched. Getting rid of the antibodies, no idea if that would work, because right now, at least on paper, we also have those seronegative ones, and you have T cells playing into action and whatnot. So it's a multicellular cascade. Whereas, for example, in lupus, it was the case that there was is very B cell and antibody heavy. When you get rid of the antibodies, everything is fine.
So do you think you will end up trying the CAR T cell therapy in rheumatoid arthritis patients?
At least for now, I would say there's no real rationale. In contrast to other diseases where we really know, okay, this is completely B cell driven. Yeah. So or at least say autoantibody driven. So in RA, for example, B cells are important, but not necessarily through their antibodies. It makes it whole complicated. So you can deplete B cells using infusions, rituximab, that gets rid of your peripheral B cells, so you don't have any B cells in your blood. In all patients, in RA patients, that has an effect. Although your antibodies don't change. In lupus, your B cells, your peripheral B cells are gone. Your antibodies don't change, and it's usually no effect. So it's always a very mixed bag.
Yeah. So, changing a bit of the topic, we talked a lot about inflammation and about the inflammation going into a loop, right? About non-stopping. And one key player in this loop, it's IL-1 beta.
Hey, you guys listened.
Yeah, of course. So, what is IL-1 beta, and what is its role in all of this loop, and why is it important?
It's one of those things that it's an immediate, I would say, alarm signal. So immune cells in your body usually have it in a form that is not active, but it can be made readily available by just cutting off a little piece. So usually if you talk about cytokines need to be produced first. So you need to kick off the machinery and get the guys to wake up and make IL-6, for example. That takes a while. That takes a couple hours. If you want to fight off, for example, some bacteria, hours that might take too long. You might want to recruit something now. And IL-1, this whole family is great because you already have it, and you just need to activate it basically. And this activation can be done by some molecular scissors, which we call inflammasomes. And this is something you can easily activate in an immune cell. Not even only immune cells. So epithelial cells can do that as well. So just by activating the scissor, which is actually you just have to make them accumulate in a complex in your cell, and then they're active. Then they activate your pro IL-1 beta, and then it's ready to go. Then it can be released and can activate all kinds of different things. So it's a very universal alarm signal, I would say, that your cells can produce, and especially the innate system. Its role as an amplificator comes from the fact that it actually feeds into the same immune pathways that, for example, other cytokines, but also, for example, bacteria-derived products entertain. So it can amplify certain signaling that is already going on. It can be a feed-forward loop in an inflammatory sense. And that's why it's so powerful.
And that is why it can be harmful. Exactly.
So if you have uncontrolled IL-1 activity, if you have out-of-control inflammasomes, that's no bueno. Do we still have tea, actually?
Yeah, I think so. No.
No!
It's over.
Okay.
Did you like it?
Huh?
Did you like it?
It was actually not. So I didn't taste the plum.
Yeah.
I could smell it.
You can smell
really?
it?
Yeah.
Maybe. Some sweetness to it, at least.
Just go to South Spain. They sell a lot.
No, so you're right. So if your inflammasomes and this whole compartment is out of control, which is often the case with periodic fever syndrome, something that manifests sometimes very early in life already, that can be quite devastating. But luckily, within our healthcare system, that can be controlled also quite well. So if you identify it, okay, you have a problem with IL-1, you can just block IL-1, and literally be done with it. Yeah, so this is a blockade you need to maintain through injections, but then basically your problems are more or less solved, which also tells you, and those patients, by the way, they are not really immunocompromised then. Yeah, so IL-1 is something where, like, okay, maybe in general we might have too much. Yeah, so it's again all about balances. Yeah, same as with the neutrophils, for example, in COVID. Yeah, so something that helped us maybe, I don't know, a couple thousand years ago, since we stand up and don't sniff at each other's behinds or whatever, we don't need this armory of innate power that self-destructs us potentially.
So what are some IL-1 driven diseases that you can treat with the blockers right now?
Well, you have all these genetic things.
Yep.
Yeah, for example, any sort of inflammasome-mediated periodic fever syndrome. You have, for example, the Mediterranean fever. This is one big group, and the other one is any sort of, for example, metabolic disease. Yeah, for example, gout. Yeah, so gout and gouty arthritis is one of the prime IL-1 driven metabolic diseases. Yeah, so here especially your neutrophils go nuts when they see certain crystals, urate crystals in your tissues, and they just produce IL-1 like crazy. Yeah, so if you can't manage your gout with conventional means, diet, no beer, no smoking, whatever, like taking the fun out of it, and then you might have an option in IL-1 blockade. Yeah, so to the point that, for example, your innate immune system can respond to all kinds of weird things that shouldn't be there. Yeah, so certain crystals, cholesterol crystals, arteriosclerosis, and stuff like that. If you were to catch it early on, like really early, maybe still in your your cohort, for me, most likely it's already too late. Yeah, so I think my IL-1 phase is already gone. Yeah, yeah. So early on, that's why pharmaceutical companies are actually quite interested in it. Yeah, so the response of the immune system to cholesterol crystals. Yeah, that's something that happens early on, and that might actually be also IL-1 driven response initially. Yeah, that then kind of develops some chronic circuits, then manifest in proper inflammation that is then IL-1 independent anymore.
So most of the autoimmune diseases start with chronic inflammation. I mean, maybe not most of them, but it's...
Well, they start with an acute inflammation.
Okay, so this acute inflammation, if it's diagnosed very early and it's treated with IL-1 blockade, can it be stopped for ended up in a worst case scenario?
Maybe, but this is really hard to address because
very futuristic. Yeah.
Well, it's not necessarily futuristic. It's more like a theoretical thing.
Okay.
Yeah. So all or most of the animal models and model systems here, IL-1 blockade can actually do something, but all the clinical trials actually failed. Yeah, so if you have an established rheumatoid arthritis, blocking IL-1 doesn't do anything. So that means either IL-1 is out of the system, it doesn't play a major role anymore, or it has lost its exclusive role. Yeah, so with the idea that actually you initiate the chronification. Yeah, but then the chronification, once everything is recruited and produces TNF and IL-6, you don't rely on that stuff anymore. Yeah, so it's a self-sustaining inflammation that is IL-1 independent. Yeah, so and that's why, at least in established autoimmune diseases, IL-1 doesn't really play a role anymore.
Okay.
Sorry.
But with something like gout, where you have episodes of it.
Yeah.
In between those episodes, is IL-1 beta still important?
Well, it's important then to that's the next hit.
Yeah.
Yeah. So if you, for example, we have patients which you treat with anti-IL-1, and if you keep treating it, they don't get gout anymore.
Yeah.
So it's again the thing with gout is that it's acute on acute upon acute. Yeah, so that you always have that neutrophil-driven response, and the neutrophils are a major source of the IL-1. As soon as you get, for example, in RA, neutrophils are not necessary to maintain the inflammation anymore once it's established. Then you have like bad macrophages, T cells, B cells going on. Yeah, so when you deplete neutrophils, then it doesn't really make a trick anymore. Mm-hmm. Yeah, so neutrophils and IL-1 go hand in hand very often. So if something is acutely neutrophil mediated, then maybe IL-1 blockade actually does something. Yeah, it's the same with, for example, multiple sclerosis. So multiple sclerosis, definitely not. You would never ever try to target neutrophils. Yeah, mm-hmm. But if you look very, very early in any sort of model, you can actually see the break of the blood-brain barrier associated with neutrophils. Mm-hmm. But that is super early, yeah, and you will never ever find those patients, because they are not patients yet. Mm-hmm. So they are patients at risk or soon to be patients. But this is how it's more. This is how it all starts, maybe. Yeah, but this is not when we see the patients.
Based on your research, do you see a future where we will target inflammation differently than how we are targeting it now?
Yes. So in principle, what we're doing at the moment is quite good. Yeah, in a way that we're targeting key molecules that entertain, let's say, vicious circuits of mm-hmm. inflammation. Yeah, so that per se is a good job. Yeah, we need to be better at understanding which circuits are actually affected. Yeah, so right now it's basically we just know from trial and error which things work and which don't. Mm-hmm. Yeah, so this whole idea of precision medicine in inflammation research is still a little bit very foggy. Yeah, so we can't tell you which cytokine blockade is needed for each patient just based on molecular profiling, for example. Mm-hmm. We just know it from historic failed trials. So we need to be better at this. Yeah, we need to understand and identify the circuits that actually are wrong. So it's not a common practice that, for example, a patient with psoriasis comes to you and you start with anti-TNF. 3 months later, did it work? No, it didn't. So, let's try anti-IL-23. Mm-hmm. 3 months later, did it work? No. Let's try IL-17. Yeah, it's like, did it work? Yes. Well, wonderful. Yeah. So next patient is we try with anti-TNF. It's like, it works beautifully. So the stratification and classification of mm-hmm. patients is still very not present. So we need to know which cytokines are involved, and then it's the question if we can identify the reason or the particular cell that mediates the disease. Yeah, for example, in lupus, if we take out the bad antigen-producing cells, apparently that's enough to get rid of the disease. Mm-hmm. Yeah, beautiful. Yeah, but as long as we don't know that, we don't know what to target. Yeah, so the problem at the moment is not actually the weapon. Yeah, it's actually the target. Mm-hmm. Yeah, so for example, in rheumatoid arthritis, no idea what the actual antigen is against which the immune system is directed and cause disease. Yeah, in lupus, you kind of know it. It's certain self antigens. Mm-hmm. Yeah, and you, in fact, now with the CAR T cell studies, we know it's some bad or some evil plasma cells going rogue, mm-hmm. and you get rid of them, and apparently that's enough, or at least for now. Yeah, so it's more like identification of the target, which we need to do. We need to do a lot better. But this is not my job or what I'm focusing on. I'm trying to understand more the concept. Okay, what can the tissue actually bring into play? Mm-hmm. How can tissues defend themselves against unwanted inflammation? And then, especially in a clinical context, is like how can we assess if a tissue is healed or not? Yeah, are some drugs, for example, better in healing tissues, to bring tissues into a state where they actually not susceptible to inflammation anymore, but actually have a kind of a resilience. Mm-hmm. Yeah, and this is, for example, just very simple, the organization of certain macrophage compartments. Yeah, when you compare it, for example, with neutrophils in tissues, they run around at a certain speed. looking for trouble. Very simplified view. Don't talk to neutrophil biologists about that. But it's like neutrophils run around through tissues, macrophages. They take, for example, 20 minutes to cover a potential site of damage. If a neutrophil is within reach and manages to get there within 5 minutes, you get inflammation. So it's a numbers game, and how much you invest into your architecture in a tissue. So it's like, okay, why don't we have more macrophages in tissues? Because they cost. You have to maintain them through growth factors, and that is basically energy and money. In those kind of a tissue economy. So economy and organization comes into play, how you build a tissue and how you then actually get back to this physiological tissue state.
So at the end, like understanding the tissue environment, how are these networks between macrophages and fibroblasts and epithelial cells and everything will lead, or at least we hope that it would lead to therapies that will be more effective.
Well, at least
to understanding how the therapy will end up working.
Yes, and to also define somewhat a target for our therapies. So it can very well be. So I wouldn't be so. One thing that you, as a rheumatologist, have to learn very early on is to say, I don't know, because most of the stuff we don't know. Because there's no answer to it. Because as I initially said, it's a huge universe, and you have those outer rims where the huts rule. This is like weird environments that no one likes to talk about and whatnot, right? And so I believe that we are still very far away from understanding everything. It's like the universe. And so having at least from a tissue perspective some sort of, okay, you're healed here. At least from a perspective of chronic inflammation, for example, psoriasis, where you don't have any T cells or B cells involved, where it's really just the inflammation. So this is where we can define, okay, you're healed. At the tissue level, because usually initially, for example, your macrophages were off or something. To develop metrics for healing.
So thank you very much for the conversation. It's very deep. And now that we have finished RT, it's the time for the last SIP question. So at the end of every chapter, we asked these 3 questions to every guest.
Okay.
So the first one is, if you could go back and speak to yourself on your first day in the lab, what would you say?
Start labeling your tubes better. Take more.
That's a very good tip.
Take more notes.
Okay. Okay.
That's great.
Okay. The second one. What's one mystery, scientific or personal, that you hope to see solved in your lifetime? Doesn't need to be immunology related.
A mystery. Well, it's a kind of a boring mystery, but like from a macrophage perspective, it's already nerdy, but it's like what I always wondered is why. It's like what defines the number of macrophages in a tissue. That's from a scientific point of view, but very macrophage heavy. And personal is like, what does my dog dream at night? Because he keeps running and punching me in his sleep.
Yeah, mine too. And then the last one is, if you could ask the immune system one question and get a clear, honest answer, what will you ask? There are 3 very specific questions, so every answer is valid. There's no correct answer.
And we can cut it, right?
Yeah, we can cut out the dead space.
Yeah.
A very specific question to the immune system. Should have prepared that, huh? Give me a second.
It was not in the mail, so don't worry.
Maybe I should ask the immune system if the hypothesis of my ERC starting grant is true.
I mean, yeah.
And if it could give me any hints for a consolidated grant.
That's actually that will give us work. So yeah, it's a very good one. So thank you very much for the talk. Yep. I think it was very nice also to have a first chapter in innate immunity. It was very good. So
thanks for coming over. Thanks for the tea.
Pleasure.
That's the point.
So stay curious
Stay critical.
and see you next time for some tea.
Bye, guys.
Reach us on Instagram at iimmunefau or send us questions, ideas, or feedback to iimmunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunkstudio and Thomas Bauernschmidt.